Right, so today we're going on to the next type of cell division, which is called meiosis. Remember, we covered mitosis, and I know some of you found it maybe a little bit tricky, but unfortunately, newsflash, meiosis is kind of worse, it's more complicated. Right? So, meiosis, which is your second type of cell division. So we're still on assignment 2, and you've got a bunch of questions in that assignment to cover this time. So just as a quick recap, right? So with mitosis, which we started, what are the purposes of mitosis? We did it very recently, right, at the start of today's lesson. So, yeah.
Grow?
Yes, so it was growth, repair,
Replace.
and replacement, exactly. Right, and you remember that those cells you're producing, are they identical to the original cells, or are they different? Identical, right? So you are essentially producing identical cells. So how is this different to meiosis? Well, does everybody know which kind of cells you produce?
Sperms.
Right, sex cells, exactly. So this one is used for one purpose and one purpose only, whereas this one had quite a few purposes. Meiosis is one purpose, and this is production of sex cells. So what do I mean by sex cells? Specifically, egg or sperm, exactly. So it will be egg and sperm. So my next question then is, how is egg and sperm different to any other cell in your body? In which way are they different? Okay.
There's 23 chromosomes.
Right, exactly. So the first difference is that you have, well, if we apply that not necessarily to human cells, but generalize this rule, so I don't know to write 23 chromosomes, necessarily. Half, exactly. So what you have is half the number of chromosomes which you would normally have in any other cell in your body, right? So half the chromosome number in sex cells. Right. Now, what I haven't written here, because I'm a bit lazy, but you could write it if you wanted to. These are the differences between sex cells and all the other cells in your body. So that's the first difference, right? Half the chromosome number. What do we call that chromosome number, which is half? There is like a word attached to it.
Haploid.
Haploid, exactly that. So that half number is called haploid number, yeah? And in humans, that number of chromosome, haploid number of chromosome, Rebecca, you already said it, was the number 23, right, for humans. And this is as opposed to the full number. What do we call the full number? Just as a like extra diploid, exactly. So I'll put it in a sort of little box here. So diploid will be full number, which is 46 for human cells. So your skin cells, your neurons in the brain, your pancreatic cells, all of those will contain diploid number of chromosomes. And it's only our egg in the female and sperm in the male, which are going to be the exceptions, which are haploid. So that's the first difference. What's the second difference between sex cells and any other cell in your body? There's another important difference. Anybody's got any other idea?
The difference between sex cells and normal cells.
Yeah. So one difference is that they contain half the number of chromosomes. What's the other difference?
They go through 2 stages of division.
Well, yeah, but that's the result of meiosis. But I'm trying to kind of get to the point of, well, once you produce those cells, what is the difference? Not in terms of how they are produced, but in terms of they're absolutely right, absolutely right. So the second striking difference between your skin cells, your brain cells, with egg and sperm is that egg and sperm are non-identical cells. So they're genetically unique. Let's just put it in a little bit of perspective, right? This feature, right? Because everybody sort of reads it and says, well, yeah, nods along. But let's just sort of appreciate what that means in real sense of the word, right? So that's something you should have done, or maybe in some groups they're still going to do it in the future. Let's talk about male and female reproductive systems, which we already started covering with your group, right? So with a female, so how many eggs are we born with? Can you remember the number, roughly? Maybe you can find it. It's a bit more than... No. You're not... 500,000. Right, exactly. So in a female, you're born with... Let me write that down as well. So born with... So 300 to 500,000 eggs, right? And how many of these eggs are we going to release? You remember we did that little calculation at one point. That's a number you would think of. How many? Around about 480, or if you want to round up, maybe around about 500 eggs we're going to release out of that mass of eggs during our lifetime, right? So you have to appreciate that initial number, this 300,000. Every single egg in every single follicle needs to be different to the next one, and 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 so on and so forth, right? So that's huge, isn't it? What about male? Remember we talked about those numbers. So how many are we talking about per ejaculation? So per...
That's a lot of sperm.
So per ejaculation, between 200 to 500, not thousands, you remember, but million.
That's what you like to talk about.
Right? So this is per go, right, as we said. Right? And again, every single one is different to the next one, and the next one, and the next one, and the next one, and the next one. Right? So isn't that amazing? That kind of mind boggles, doesn't it? That your body has to produce that kind of variety of outcomes where every single sex cell is different. And we can also relate to that fact by thinking about us, and your parents might have siblings, you might have siblings. Anybody's got siblings? Mm-hmm. Yeah, okay. Now some of you might say, well, I'm pretty similar to my sibling, but can we still tell you apart? Say, this is Corey, and this is Corey's and a brother sister, right? What was it? Brother sister. Yeah, right, sister, right? Are you similar to each other, or are you fairly different? Different, right?
Yeah.
The same to parents.
No.
No, okay. Well, yours is a little bit maybe different. So at least some of you.
I look like my brother, but
Yeah.
we've got the same parents.
Right.
It's good
But
to
even
like
if
that,
one of your parents is the same, but other parent might have been different, so it gave you a bit more variety. But if we take the same set of parents and they have kid after kid after kid, right? You can appreciate that often in the families it is the case that the siblings are very, very different, right? So it might be different heights. One has got blonde hair, another got brown hair. It might be different eye colors, different inclinations, talents. And even if they are fairly similar, we can still tell them apart. Right? And all of this comes from the fact, even though we don't sort of think about it very often, but you started with the same set of parents. They haven't changed. Their genetic makeup hasn't changed. But they managed to produce completely different kids with different sets of features. And this is all due to meiosis, ultimately, right? If meiosis didn't exist, if we all just relied on mitosis, well, all of us would be cloned, right? We'll all be sort of however many billions of Chris's just walking around, right, on planet Earth. We're exactly the same.
Won't it be that, like, there won't be boys and there won't be girls? Do you know what I
Yeah,
mean?
so then in that case, if you're relying on a type of division where you are genetically identical, then you're right, it'll be a bit like bacterial cells, because that's what they do, right? So they don't have boys and girls, there isn't boy bacteria, girl bacteria, they just keep on dividing irrespective of their sex. So they're relying on asexual reproduction, whereas we rely on meiosis, which is sexual. Right. So can you see that these differences mean that meiosis you're trying to achieve a lot more compared to mitosis, and then that in turn means that meiosis is more complicated, right? Which is what I said, right? So it's a more complicated process. So can I wipe all of this off? Okay. So one of the things in which, one of the ways meiosis is more complicated just from the start, it relies not just on a single division, but actually you have to divide twice in order to get to the end product of meiosis. So in mitosis we had our parent cell that divided and you ended up with two daughter cells, the end, right? With meiosis it's more complicated. So you start off with your parent cell, and that contains 46 chromosomes. Then you divide it once, and this one each still contains 46 chromosomes, but you'll see that those chromosomes change somewhat, right, to make them different to the ones you started with. And that first division is your mitosis one. Let me just have a think, actually. Am I making a mistake here? I might be making a mistake.
Why would it be mitosis?
Yeah, I think it's 23 still, sorry. You're already going to 23, but each of those chromosomes contain two sister chromosomes. That's how it works. Sorry, got a bit wrong here. So that's the correct one.
And that's for meiosis or
So
mitosis?
this is meiosis, yeah. So this is meiosis
yeah.
one. See, when you try to do recording and try not to make any mistakes, so it's more, so you end up with something like that. And then the second division, where you end up with your full outcome, so let's say these are sperm cells, and each of these is going to contain 23 chromosomes as well. And that's going to be your second division, which is your meiosis 2. That's better. So here you started with 46 and you ended up with 46. So each of those divisions, it has, again, stages, and the names of stages are the same as we had in mitosis. So in meiosis 1, in this first division, and usually we use Roman numeral, you will also have, what's the first stage normally called? Can you remember? Right, that's before
Prophase.
division. Prophase, yeah. So prophase, and then to identify that that's part of the first division, you just take Roman numeral 1 at the end of it. Then after prophase, it's which one? Meta. If you don't mind, I'm not going to write all the phases, so just put pro meta, so still 1. Next one is Anna. And one after is Teller. And then that's going to be followed by cytokinesis. And then the same thing is going to happen then in meiosis 2. So you're still going to have your pro, but this time you have 2 after everything. Meta, Anna, Teller, 2 and cytokinesis. I'm just using a bit of shorthand, but in the assignment make sure you put full names of stages, not just meta, Anna. I'm just trying to save a bit of time. And then before meiosis 1, you will have interphase. But then you will not have interphase between first and second division, so no interphase here. And so when you get all the way to cytokinesis 2, this is when you're going to end up with your end product, which is either going to be sperm or egg. So far, so good? Yes? So what we're going to then start doing is looking at each stage one at a time. So the longest and the most complicated one is this one, prophase 1. So once we have that one out of the way, then it'll get hopefully easier. Can I wipe all of this off again to free up a little space? Yeah, a bit. Yeah. So a lot of the events in those stages are very similar to mitosis, but there's going to be some things, some events which happen in meiosis which did not occur in mitosis at all. So let's talk about interphase first. So this is very similar to mitosis. What would normally be happening in interphase?
DNA replication.
Okay, exactly. So DNA replicates. And that means you remember that each chromosome in that is then going to have 2 sister chromatids as a result of DNA replicating. Right? And apart from that, so the DNA replicates, what else normally replicates, you remember, in interphase? Organelles, yes, you have organelles.
So is it the same for meiosis one?
Yeah, so as I said, a lot of it is similar. So you'll see a lot of similar events happening from one stage to the next. And then other than that, again, cell just living its life, right? Sort of normal functions. So once the cell is ready, it's then going to go into the first division. You remember that the original cell, the parent cell, it contains 46 chromosomes, so diploid number, not 23. So it's like a pretty much bog standard normal cell of your body. Right, so then we're going to go into the first division, so meiosis 1, and the first stage of that division is going to be prophase 1, right? So what's going to be happening in prophase 1? Again, let's think about what would normally be happening in mitosis in prophase. What are the typical events of prophase?
The nuclear cell membrane.
Okay, nuclear membrane disintegrates. Okay, so let's note that. So nuclear membrane disappears. What else normally would happen?
Condense.
What condenses? Chromosomes condense, good. And what else happens normally? One more.
Spindle.
And then spindle starts to form. Okay, spindle. So all of those things you're used to, right? So you should be familiar with those events from when we talked about mitosis. But there is one extra process which happens in meiosis which does not happen in mitosis. And this process is called crossing over. So I'm not going to illustrate all of those previous processes because, as I said, we talked about those, so you should have a fairly clear idea what they look like. So let's just think about that one, crossing over. So to illustrate crossing over, we will need some colors. So you need 4 colors of pens or pencils or felt tips. So I do have a pot of felt tips. Or if you're watching from home, just grab yourself some colored pencils or pens or highlighters, whatever you want. It doesn't really matter which colors they are. So I think we need a couple, actually 2 colors will do.
I'm confused now.
Yeah, what is it? Confused.
Why would meiosis 1 yield 23 chromosomes and not 46?
Right, so I think the answer to that is going to be answered in metaphase 1. So wait until we get to metaphase 1 and you'll see why this is the case.
No, I get it, but if it's already separated into 23 chromosomes.
Yeah, because again, if you wait until we get a bit further, but in a nutshell, because at the end of the first division, each chromosome still, even though you ended up with 23 of them, but it still contains 2 sister chromatids. So you need to still separate those. Okay? Wait until we get to the end of the first division.
No.
It'll make more sense.
Correct in my head.
I get what you're trying to do.
Yeah, I understand what you're asking, but
Yeah.
wait until we get to that. So crossing over. So I'm going to use, I don't know, which color shall I use? I'll use green and red, maybe?
Can I just clarify something?
Yeah, go on.
Is crossing over what all that's called, or is crossing
No,
over another?
crossing over is one of the processes which
Another
takes
process.
place in prophase of meiosis 1. And this is a
It
new
doesn't
process
happen in
which didn't happen in mitosis because
Okay.
all the other things, like nuclear membrane disappearing, etc., that was all happening in mitosis. So that's why I'm not sort of talking about those in detail, whereas crossing over, well. Maybe I'll do blue and red. Yeah, but it doesn't really matter which colors, just use your favorite colors. So I'm going to draw one of the chromosomes in one color in my diagram, and my second chromosome is going to be a different color. So these 2 chromosomes contain 2 sister chromatids. But these 2 chromosomes I've drawn, they're so-called pair of chromosomes. So this represents a pair of chromosomes. Now, you have to tell me, you remember, I think we covered that fact, that you've got 46 chromosomes or 23 pairs. So why do we organize chromosomes into pairs? Why don't we just say 46 chromosomes, full stop?
One from mum and one from dad.
Right, exactly. So this is just a quick reminder, so I'll draw it as a separate little picture on the side. So what you have is, you've got your 46 or 23 pairs, and what Emma was saying, they're pairs because within each pair, one came from your mum and another came from your dad. Do you remember that? Right, so that was in a little lecture about terminology of introduction. Can anybody remember though, or looked up in your notes, what do we call, there's like a technical term for the chromosomes which belong to the same pair? What's the word given to it? Have a look in it, see if you can find it. So what do we call two chromosomes which came from the same
Homologous?
pair? Let's see if anybody else can find it.
No.
Yeah, okay, good. Have you all found it? Yep. So we call it homologous pair of chromosomes, right? Homologous. Why are they in a pair? Because, and again, this is something which was covered in that introduction to Meiosis lecture. They are grouped in a pair because whatever feature is being coded by one chromosome, which came from your mom, let's say your ear shape, the same feature, ear shape, also would be in the same place on your dad's chromosome. But the actual code might be different. So this one might be saying, big ear, this one might be saying little ear. But they are both coding for the ear shape, nevertheless, right? So they code, homologous chromosomes code for the same set of features. So if you have a different pair of chromosomes, they might be coding for something else, right? So the important thing to remember, this process of crossing over, that takes place between homologous chromosomes. So you can't take different chromosomes from different pairs and do that process, because that would lead to mutations. Right? So it's got to be a pair of homologous chromosomes. So these chromosomes, they find each other, the pairs find each other, and they literally lie on the top of each other like that, right? Like two pancakes on top of each other. I can't draw it like that, that's why I'm drawing it side by side. But you have to imagine that that's how they happen. So they really get super close and get on top of each other. And in crossing over, what's going to happen is that these chromosomes are going to start swapping bits between each other. So that's what I'm using colors for, to show that swap. So let me see if I can figure it out. So this one, let's do red like here. And a little bit of a red like here. And this one. Okay, so how we draw it. Bring this with me. So something like this. And this one. Just straighten it out. It's going to be wonky. Okay, I hope that makes certain sense. Right, so those bits which they exchange, they need to be equivalent in size. You can't exchange bigger piece for a smaller piece. So what I was trying to draw is one kind of slightly bigger section getting exchanged on here, and slightly smaller bit getting exchanged here. picture makes sense. Okay, so all I've done is broken off a piece of homologous chromosomes. Well, let's say blue one is from your dad and red one is from your mom. So that blue section from your dad is then now attached itself to your mom's chromosome, and that bit went across.
Both.
Right?
You have
Yeah.
X and
So
Y.
then the same happened to this other leg. Yeah. So whatever bit broke off with that blue chromosome that went across, the equivalent bit then attached itself to your dad's chromosome from your mom. So that's what you have. So those chromosomes, they're a bit of everything now, right? A bit of your mom, a bit of your dad, and each one. Now let's figure out what is being exchanged exactly. Are we exchanging bits of sister chromatids or non-sister chromatids?
No.
Right, absolutely right. So it's non-sister chromatids which are getting exchanged. So again, going back to that picture, let me just free up a bit of space here at the bottom. Yeah. So if I draw arrows pointing to this and this bit, what are these two bits called? Sister chromatids, absolutely right. So sister chromatids.
That's cool.
And what if I point to this leg and this leg?
Non-sister.
No.
Then these are non-sister chromatids, right? So they are non-sister. So you've got to be careful with that bit of terminology as a result, right?
So that's how you get this from you, this from you.
Right, exactly. So what you're exchanging is sections of non-sister chromatids which belong to the same pair of chromosomes, right? Because if I exchange sister chromatids, well, I would be exchanging bit of blue for another bit of blue. That wouldn't have, led to anything.
It's gonna look like that.
It would be the same as what you started with. But when you're exchanging it between two different chromosomes, that's what makes all the difference.
Is that why sometimes you can look more like your mum than your dad?
Absolutely right, right? So, or your
And you can look like that.
child can look more like your granddad compared to your grandmother, etc., etc., right? Because, you might have got bigger portion of the chromosome which came from, your grandparents, right? Because these chromosomes originate from the grandparent of a child who's going to end up being part of that sperm or the egg, right? So you can see this one has got more information from your dad, this one has got more information from your mom, but it's still a bit of a combination. And then another important thing to remember about crossing over, that this process is completely random process. Right? So you don't know in advance which parts of these chromosomes are going to get exchanged and how many of those parts are going to get exchanged. So it could be, like I've drawn here, a couple of fragments getting exchanged, but equally it could have been just one, it could be 5, 10, it could be hundreds of really tiny fragments. And you can't tell what's going to happen. This is just purely, random process, right? So can you now see how that can then lead to non-identical cells, that crossing over? Because those chromosomes never existed before. You just took and really, rearranged all these genes in sort of random way. Okay? Is that fairly clear, right? So that's the extra process you need to talk about in your assignment when you get to prophase 1. Okay? Moving on? Yeah. Wiping it all off. Right, so after prophase 1, what's going to be the next stage?
Metaphase.
Metaphase, which number, 1 or 2?
One.
With 2, are we already on second division?
No.
No, we're still on the first one. So the next one is going to be metaphase. Metaphase 1. Now, can you remember what was happening in metaphase in mitosis?
The chromosomes were aligned.
Well aligned, do you remember? So the key feature of metaphase is things or chromosomes aligning in the middle. So this is what's going to be happening in metaphase 1 as well. However, one difference is that it's not going to be just single chromosomes aligning, but rather we're going to have pairs of chromosomes aligning. So that's right, so pairs. And those pairs are still the pairs of homologous chromosomes from previous stage. So they align at this middle point of a cell, which we also which is also known as equator. So we're going to draw another diagram, if you don't mind. You know what I'm like with diagrams. And for this one, we will need 4 colors. So keep your first original colors, which you had for crossing over. We'll need to add 2 more colors. So I'm going to draw the first picture here. So that's going to be my middle of the cell. And that's going to be my spindle fibers. Okay. So now I'm going to draw my pairs of chromosomes. So my first pair of chromosomes, you remember I used red and blue, so I'm going to stick with red and blue. Now, here is the thing which is a little bit confusing. If you don't mind, instead of drawing crossing over which happened in the previous stage, do you mind if I just skip one chromosome blue and another one red? But just keep in mind that they crossed over. I mean, I could have, if you insist, I could do it like that. But that's going to make things even more complicated, right? Can we just sort of stick with just blue and red, but keep in mind that, as I said, crossing over did occur. So I'm going to unfortunately wipe it off, so not to confuse you even more. Let's just stick with these 2 colors for the first pair. And then we'll draw another pair of chromosomes underneath. And you have to remember that the same is going to be happening for your 23 pairs. But if you don't mind, I'm not going to draw with 23 pairs. Right? Is that okay? Yeah? We're good with that. So let's say, I'm using green and black. So this is pair 1. This is pair 2. And here we are aligned in the middle. Can you see the difference between this picture and metaphase of mitosis? If you are not sure what the difference is, go back to your notes on mitosis and have a quick look what the picture would be like. So here, just as a reminder, we're lining up pairs of chromosomes, right? In mitosis, quick picture, right? We were aligning individual chromosomes. It was more like that, yeah? Can you see? That's the difference between the two. And here is another thing which is important to understand in order to be able to explain where variation comes from, why each sex cell is different to the next, right? Because these chromosomes could have aligned in this way, the way I've drawn, but equally they could have aligned differently. So that's my second picture. So I'm going to keep the first pair, the top pair, the same as they were here. So my blue and my red. But this one, I'm going to draw it slightly differently. So let's see if you can pick up on that difference. So this is my pair 1, this is my pair 2. What's the difference between this picture and this one?
It's flipped.
Right? Can you see what happened? What's flipped?
Chromosome.
Which
pair.
one?
The green one.
Right, the second pair, can you see? So all I've done is I've flipped them around. So this time, on this side I have blue and green, and in my second version of this diagram, I've got blue and black now, right, grouped together. And what you have to remember, what's going to happen, right, ultimately, I'm going to draw these chromosomes to one side and these two to the other side. So in this scenario, blue and green are going to end up being together in one cell, and in this scenario, it's blue and black are going to end up together. So you can see that these two diagrams are going to lead to different outcomes. So this is outcome 1, and this is outcome 2. So these two, can you appreciate that these two diagrams would lead to different end result? Can we see that? Yeah? So don't forget, this is happening not just with two pairs, one pair of chromosomes, or two pairs of chromosomes. This is happening with 23 pairs of chromosomes. Do you remember that in real life? But I can't be bothered to draw 23 pairs. You can if you wanted to, right? But I'm not going to. So therefore, right, the more chromosome pairs you add, the more possible outcomes you're going to end up with. So can we do a little calculation to figure out how many outcomes that would be, right? Just a tiny bit of math, like this much. So there is a mathematical way of expressing it. So if you have two chromosomes, yeah, and you just kind of have them in one pair, right? So two chromosomes, one pair. Let's say I just had well, let's take this one, red and green, right? I know, right? Yeah, that's it. Need to have lunch now. Green and black. So I can have it in that position, or I can flip it in the second position. So a single pair of chromosomes, you can have two possible outcomes. If I have still two chromosomes in each pair, but I've got two pairs of chromosomes. How many possible outcomes? 4. 2 in the power of 2? 4, right? It's just 2 multiplied by 2, right? 4 outcomes. If I have still two chromosomes in a pair, that hasn't changed, but now I'm playing with 3 pairs of chromosomes. How many outcomes is that going to be?
9. 2 times.
Right? So
no, 6.
So 2 times,
8.
so it means
8.
2 times 2 times
8.
2, right?
8.
Yeah, that's
Sorry.
what that means.
8.
So it means it's 8. I really have to do that. Alright, and next I would like you to find your calculator, use your phone or your scientific calculator if you have one. And you remember, we don't have 1 pair or 2 pairs or 3 pairs of chromosomes. How many pairs do we have? 23. So it's 23, 2 in the power of 23. So you have scientific calculator, you can do it straight like this. If you don't, if you're just using phone, do it 2 multiplied by 2 multiplied by 2, 23 times. So let's see how many we're going to come up with. Okay, so can you have a go? I can't do that now.
I'm gonna get my calculator.
Yes, please
do.
Googly. Google.
Watch it now, batteries. I haven't got batteries.
Just use your phone. then.
Two.
Oh, yeah.
Oh my god.
This is
I can tell you've not used that now, have you?
8,000.
2 to the power.
Which number is this? 8388608. So you go.
No, what are you on?
We're sure we're on the same page. Chris, are you doing yours? You wish somebody else to do yours?
I've got
Okay, have a go.
8388608.
So do you both have the same number?
Can you show that to our camera?
It's
8,388,608.
You need to know how you calculate specifically.
Your calculator's different.
So that's a long number. I
Just
can't
do
say
it
that.
2 by 2 by 2 by 2, just
Ah.
keep track. of
See, like mine's got that.
So how many, how much have you?
yeah.
Okay. And
So
yours is the
it's
same,
a
yeah?
Power of 23 look like that.
Okay. What do we have?
I don't know.
Oh,
That'll be
yeah,
useful.
you're not gonna have, yeah, so you'll have to just do 2 by 2 by 2 by 2 by 2. How many twos you want?
608 on them,
Right, so I think we've got a couple of people with the same answer, so let's trust that they got it right. Right, how many have we got? Rebecca, do you want to read out?
838.
838, yep.
Yeah, 8608. 838, 8608.
Like this. Yeah?
Yes.
Yeah, that's
This,
it.
yeah? Okay. Right, so what is it? Is it thousands? Is it hundreds of thousands? Is it
Millions.
millions? Millions, absolutely right. So we've got round about 8.4, right? Million outcomes. Right? So what that means is that even if I disregard crossing over, by just flipping these chromosomes around during metaphase 1, I could still end up with over 8 million outcomes. So if you combine that process with crossing over, then you get literally unlimited number of variations. And that's why, you know, even though males produce millions of sperm per ejaculation, they can still end up with every sperm cell being different. So this process, what I've drawn here, it's got a bit of a convoluted name. It's known as independent assortment of homologous chromosomes. Can you write it down?
How? Independent. Associate.
So this is known as independent assortment. Independent means, you know, you don't know which chromosomes are going to end up on which side of the equator. Assortment, a bit like licorice, all sorts, right? You're just picking whichever happens to be on that particular side of the equator. And we're doing it with homologous pairs of homologous chromosomes, so independent assortment of homologous chromosomes.
Yeah.
You've got these different, end up with different outcomes.
So the outcomes are independent of something.
Well, the outcomes, that number of outcomes is created by this process called independent assortment. And
Okay.
independent assortment was the fact that you can just flip those chromosomes within each pair in a random way. You don't know which chromosome out of each pair is going to end up on which side of the equator. So these two processes, independent assortment and crossing over, they both contribute to each sex cell being different, to variation. Right, so we're going to stop at this point and we're going to carry on with the rest of the stages, right, anaphase, etc. in the next lecture. Right, do you want to